Welcome to Lesson 17.2, Evolution as Genetic Change in Populations. In this lesson, we'll explain how natural selection affects single gene and polygenic traits. We'll describe genetic drift, and we'll explain how different factors affect genetic equilibrium. When a pesticide is introduced, like the one shown in this picture, it typically kills most insects. In evolutionary terms, the pesticide becomes part of the insect's environment that dramatically reduces the fitness of most individuals. But usually, just by chance, some individuals in the insect population carry genetic variation that enables them to resist the pesticide. Pesticide resistance is an adaptation that enables individuals to survive and reproduce. So what happens when a pesticide is introduced into an environment? The presence of the pesticide increases the relative fitness of the few individuals that carry the resistant adaptation. Resistant insects survive and reproduce, passing resistance genes to their offspring. Meanwhile, the pesticide eliminates reproduction by individuals that don't carry the resistant genes. Over time, the frequency of a resistant allele increases in the population. This allows us to define evolutionary fitness in genetic terms as an individual's success in passing genes to the next generation. An evolutionary adaptation is any genetically controlled trait that increases an individual's fitness. When natural selection produces differential reproductive success, the effects on phenotype can look different for single gene and polygenic traits. Imagine, for example, that a population of brown lizards experiences mutation in a gene that determines body color. The mutation produces red and black forms as shown in the table. So what happens to the allele and phenotype frequencies? Well, if red lizards are more visible to predators, they might be less likely to survive and reproduce. Therefore, the allele for red coloring might not become common. Black coloration, on the other hand, might enable individuals to absorb more sunlight and warm up faster on cold days. If high body temperature allows individuals to move faster when feeding and avoiding predators, black coloration can act as an adaptation that increases fitness. In that case, the allele for the black coloration and the black phenotype would both increase in frequency. If black coloration has no effect on fitness, the phenotype produced by the mutant allele will not act as an adaptation. In that case, the allele will not be under pressure from natural selection. So what's happened to produce the population shown in generation 30? Well, red lizards have been selected against because they are more visible to predators, while black lizards have been selected for because they can move faster and avoid predators. As a result, red lizards have been eliminated and black lizards have become more common. When traits are controlled by more than one gene, both the trait and the effects of natural selection on the trait are more complex. Polygenic traits, such as height, often display a range of phenotypes that form a bell curve like that shown. The fitness of individuals may vary from one end of such a curve to the other. Where fitness varies, natural selection can act in one of three ways. Natural selection on polygenic traits can affect the relative fitness of phenotypes and produce three types of selection: directional selection, stabilizing selection, or disruptive selection. When individuals at one end of the curve have higher fitness than individuals in the middle or at the other end, directional selection occurs. The range of phenotypes shifts because individuals with adaptations that result in higher fitness experience higher reproductive success. Let's examine how a finite supply of resources, such as food, can produce differential reproductive success in this kind of population. Seed-eating birds with big, thick beaks can feed more easily on large, hard, thick-shelled seeds. Suppose the supply of small and medium-sized seeds runs low, leaving only larger seeds. Under these conditions, larger beaks would serve as an adaptation because big-beaked birds could feed more easily than small-beaked birds. Big-beaked adaptations produces higher fitness and greater reproductive success. Over time, the average beak size of this population would probably increase. If large seeds became scarce and there were more small seeds available, how would this affect the curve for directional selection? The curve would probably shift back to the left. If individuals near the center of the curve have higher fitness than individuals at either end, stabilizing selection takes place. The center of the curve remains at its current position, but it narrows the curve overall. Birth weight among human infants is a trait related to fitness. Very small babies are less likely to be healthy and are thus less likely to survive. Extremely large babies are more likely to experience difficulty being born. The fitness of smaller or larger babies is therefore lower than that of an average-sized baby. As a result, human birth weight is under the influence of stabilizing selection. So if medical advances led to larger babies having a better chance of surviving, how would this affect the curve for stabilizing selection? The curve would shift to the right. When phenotypes at the upper and lower ends of the curve have higher fitness than individuals near the middle, disruptive selection can occur. Disruptive selection lowers the fitness of intermediate phenotypes. If the pressure of natural selection is strong and lasts long enough, the single curve can split into two. In other words, disruptive selection can create two distinct phenotypes, increasing diversity within the species. Suppose a bird population lives in an area where medium-sized seeds become less common than large and small seeds. Birds with unusually small or large beaks would have higher fitness. As shown in this graph, the population might split into two groups, one with smaller beaks and one with larger beaks. If more medium-sized seeds become available and small and large seeds become scarce, how would this affect the curve? The curve would shift back to having one central peak. Natural selection is not the only evolutionary mechanism that can change allele frequencies. In small populations, individuals that carry a particular allele may leave more descendants than other individuals leave just by chance. Over time, a series of chance occurrences can cause an allele to become more or less common in a population. This kind of random change in allele frequency is called genetic drift. Sometimes a natural disaster, such as a storm or flood, can kill many individuals in a population. The gene pool of the surviving population may have allele frequencies that differ from those on the original gene pool just by chance. If the reduced population later grows, its allele frequencies will also differ from the original population. The bottleneck effect is a change in allele frequency following a dramatic reduction in the size of a population. A severe bottleneck effect can sharply reduce a population's genetic diversity and thus decrease diversity within the species. Genetic drift may also occur when few individuals colonize a new habitat. Though these founding individuals may carry alleles that differ in relative frequencies from those of the main population, just by chance. The new gene pool, therefore, starts out with allele frequencies that differ from those of the parent gene pool. This situation, in which allele frequencies change as a result of the migration of a small subgroup of a population, is known as the founder effect. These two small groups from a large diverse population could produce new populations that differ from the original group. One example of the founder effect is the evolution of several hundred species of fruit flies on different Hawaiian islands. All those species descended from the same mainland fruit fly population. However, species on different islands have allele frequencies that are different from those on the original species. Why are the two populations of descendants shown so different from each other? The two populations are different because they were founded by small subgroups of the original population that were, by chance, genetically very different from each other. The founder effect may be especially likely to occur following a major change in the environment, such as a forest fire, landslide, volcanic eruption, or human destruction of habitat. One way to understand how and why populations evolve is to imagine a model of a hypothetical population that does not evolve. If a population is not evolving, allele frequencies in its gene pool do not change, which means that the population is in genetic equilibrium. However, any wild population, like the seals shown, is unlikely to remain in genetic equilibrium. Gene shuffling during sexual reproduction produces many gene combinations. But a century ago, researchers realized that meiosis and fertilization by themselves do not change allele frequencies. So hypothetically, a population of sexually reproducing organisms could remain in genetic equilibrium. The Hardy-Weinberg principle states that allele frequencies in a population should remain constant unless one or more factors cause those frequencies to change. The Hardy-Weinberg principle makes predictions like Punnett squares do, but for populations, not individuals. Here's how the Hardy-Weinberg principle works. Suppose that there are two alleles for a gene, A, the dominant, and little a, the recessive. A cross of these alleles can produce three possible genotypes: homozygous big A, heterozygous with big A, and homozygous little a. The frequencies of genotypes in the population can be predicted by the equation shown, where P and Q are the frequencies of the dominant and recessive alleles. If this population is in genetic equilibrium, what are the chances of an individual in the next generation have a genotype big A, big A? If this population is in genetic equilibrium, what is the probability of the genotype little a, little a? And if this population is in genetic equilibrium, what's the probability of having the heterozygous genotype big A, little a? If a population doesn't show these predicted phenotype frequencies, evolution is taking place. The Hardy-Weinberg principle predicts that 5 conditions can disturb genetic equilibrium and cause evolution to occur. In genetic equilibrium, individuals must mate at random, but individuals often select mates based on size, strength, or coloration, a practice known as sexual selection. When sexual selection occurs, genes for traits selected for or against are not in equilibrium. Female peacocks, like the one shown, choose mates on the basis of physical characteristics such as brightly patterned tail feathers. This is a classic example of sexual selection. Genetic drift does not usually have major effects in large populations, but can affect small populations. Evolution to genetic drift thus happens more easily in small populations. Changes in allele frequency can be produced by gene flow, the movement of genes into or out of a population. Individuals who join a population through immigration may introduce new alleles, and individuals who leave through emigration may remove alleles. If allele frequency in the population changes, gene flow has caused evolution to occur. Mutations can introduce new alleles into a gene pool, changing allele frequencies and causing evolution to occur. If different genotypes have different fitness, natural selection will disrupt genetic equilibrium and evolution will occur. One or more of these conditions usually holds for real populations. So, most of the time, in most populations, evolution happens.